There is currently no cure for Ebola, and scientists still have much to learn about the virus. Patients are typically given broad-spectrum antibiotics, painkillers, and an IV drip in regions where these supplies are available. They are also commonly given malaria drugs, as that disease is so prevalent in areas also affected by Ebola. Early treatment is crucial, as any boost to the immune system in these stages will help the system to overcome the virus before the virus overcomes the system. Patients who survive typically develop antibodies 6 to 11 days after first exhibiting symptoms. When victims are in later stages of the disease, attention is given to organ systems as they begin to fail, for example, dialysis to combat renal failure. Again, in the regions where Ebola occurs, these options are often not available. When Ebola is suspected, samples are collected and sent to health authorities for testing. Here is what the Centers for Disease Control have to say about their testing methods. Antigen capture enzyme-linked immunosorbent assay, ELISA testing, IgM ELISA, polymerase chain reaction, and virus isolation can be used to diagnose a case of Ebola within a few days of the onset of symptoms. Persons tested later in the course of the disease or after recovery can be tested for IgM and IgG antibodies. The disease can also be diagnosed retrospectively in deceased patients by using immunohistochemistry testing, virus isolation, or PCR. Ebola continues to be an issue in Central Africa, with 11 outbreaks occurring since 2000. The most recent confirmed outbreak occurred in Uganda in 2012, and although it was contained to 24 people, 17 of them died, making the mortality rate for that outbreak more than 70%. Scientists have been searching for the natural reservoir of Ebola since the first outbreak in 1976. An extensive survey of animals found in and around the cotton factory where the first outbreak of the Sudan Ebola virus occurred found no trace of the virus present in any of the samples. Due to the prevalence of the virus in primates, they are often misconstrued as the source. However, a natural reservoir will typically be an asymptomatic host. A study of hundreds of plant and animal species in Central Africa found traces of the virus in 3 species of fruit bat, and it was confirmed that there were fruit bats living in the cotton factory in Nizara. Although there is no definite link between bats and Ebola, the current theory is that the fruit bats carry the virus and spread it when they drop their fruit. The fruit is subsequently eaten by primates such as chimpanzees and gorillas, as well as a small type of deer called a duiker. This disease is far more prevalent in gorillas than humans, and is believed to have killed more than 5,000 in outbreaks occurring between 2002 and 2004. Scientists believe the virus, in combination with hunting, will likely cause the species to go extinct in the near future. Researchers have traced the roots of several recent outbreaks to people eating tainted meat from duikers as well as primates. Here is a map that shows some of the links they've made between areas where the virus is prevalent and areas where fruit bats are found. There are several factors that cause outbreaks to occur and spread rapidly. One is the general expansion of the human population causing close living conditions. Funerals are a major contributor. One outbreak, which occurred in 2008 and infected 217 people, was traced back to a large funeral for 2 village chiefs. Lack of proper medical supplies is another major concern, and often a large percentage of victims are medical workers who do not have access to proper protective equipment. The eating of tainted meat continues to contribute to the spread of the disease as well. Since being identified as a Class 1 bioterrorism substance, a lot more research has gone into curing the disease. A drug is currently being tested that blocks the receptors on cells that allow the virus to enter the cytoplasm. A vaccine has been developed using antibodies from a range of animal bloods as well as human bloods, which achieved mixed results in lab tests. Scientists are working to discover ways to simply slow down the virus so the immune system can regain control. It would be ill-advised in every way to attempt to work on a case with Ebola. Funeral customs in Central Africa emphasize touching the body, and there is extensive evidence that this has led to the spread of the virus in some outbreaks. In many cases, funeral rites have been forgone, and bodies are buried in mass graves by groups known as corpse disposal squads. Ebola is spread through contact with bodily fluids and is known to be highly contagious both before and after death. The first human case of Typhorus Ebola virus occurred after a zoologist performed a necropsy on an infected chimpanzee. If one were to attempt embalming despite all these risks, drainage and distribution of fluid would be extremely challenging, if not impossible, due to extensive breaking of blood vessels and the massive amount of coagulation experienced by victims. Major restorative work would also be necessary due to the myriad discolorations and skin conditions caused by this disease. There can be no doubt that Ebola is one of the deadliest diseases currently infecting human beings. Outbreaks continue to plague Africa. In fact, just today, a news story reported that a mysterious disease has killed 23 people in Equatorial Guinea. It is believed that this disease is Ebola. If this is confirmed, it will bring the total number of outbreaks to 12 since 2000. There is much to fear from a virus so young and virulent, but there is also more research being done every day to prevent future outbreaks and find a cure. Thank you for watching my presentation.